This video lecture is over Chapter 24, the immune system. As always, please refer to the Chapter 24 study guide that outlines the learning objectives and the reading material that you should be familiar with and over which you'll be tested. The immune system consists of three parts: our lymphoid tissue, our immune cells, and the chemicals that coordinate and execute those responses. And these three parts work together to satisfy and complete the functions of our immune system, which are chiefly first to remove and recognize abnormal cells. So any cell that is no longer functioning within its normal cell cycle, the body needs to identify it and remove it. And that's the function of the immune system. Secondly, to remove out dead or damaged cells. And third, it protects the body against disease-causing invaders, or what we call pathogens. And these include bacteria, viruses, protozoans, multicellular parasites. And we protect against this with what we call immunogens. These are compounds that trigger an immune response. One particular type of an immunogen is an antigen that interacts with a component of the immune response. When the immune system doesn't function correctly, we have a pathology. And we can classify pathologies of the immune system in one of three ways. First, we can have incorrect responses, like we recognize and we attack healthy cells instead of unhealthy. Typically, this is what we would call an autoimmune disease. Example of that is type 1 diabetes, in which we fail to produce insulin because the body attacks beta cells in the pancreas and destroys them. Secondly, we can have an overactive immune system in which the response far outweighs what a normal response would look like. An example of this would be our allergies, right? We have allergies because the immune system is attacking chemicals that come into the body that don't really cause us any problems, but the immune system thinks that perhaps they would. And then we can have failed responses altogether. These would be called immunodeficiencies. We can have either primary immunodeficiencies in which the body just is incapable of producing certain cells or certain responses, generally that it's genetic in nature, or we can have acquired immunodeficiencies. An example of this would be like AIDS, right? If you contract the HIV virus, that can disrupt our immune system, and we then have an acquired immunodeficiency. The way that the immune system then responds to pathogens it can be classified in really two ways. First, the first line of defense of any pathogen is going to be a physical barrier. This is why we have our skin. The epidermis serves as a physical barrier to pathogens, to bacteria, viruses. Okay, we also have mechanical and chemical barriers as well. So the mechanical would be the flushing mechanisms in our body, like the cilia on the surface of a lot of cells that kind of brush off pathogens and don't allow them to attach or enter in through the membrane. We have chemical barriers, like in the stomach, right? We have the gastric acid that produces a lower pH to kill bacteria that have entered in our GI tract. Okay, that's the first line of defense. But if that barrier fails, then we have two types of immune responses. The first is what we call innate immunity. This is a very rapid, it's also non-specific, and there's not really any memory retained of the infection. The types of cells involved in this are our macrophages, our neutrophils, our natural killer cells, dendritic cells, eosinophils, and our basophils. I'll talk about these in just a minute. And then the chemicals that we have, we'll have complements and interferons and a couple others that play a role in it. And the way that these cells work is they have very broad specificity. So they recognize what we call pathogen-associated molecular patterns, or PAMPs, that are found in bacteria or viruses, and the response tends to be inflammation, and then we use cytokines that mediate the additional responses found within there. The next type of immunity that we can have is acquired immunity. And this occurs when we actually start getting very specific immune responses and we use antigens to direct that response. This is much slower. It's usually going to take a few days for this to start. However, there is memory that's retained. So we create antibodies, and these antibodies can be retained in the body. And then when we have a secondary infection, we can have a very fast response that is specific for that. And we call this cell-mediated versus humoral, so which would be what we kind of had in the innate. When we have a immune response, there's 4 basic steps that occur. First is that we detect and identify the foreign substance. After the body has identified it, then it can communicate with other immune cells. And after that communication is to recruit assistance of those immune cells and then to coordinate the response of what should be then done with that foreign substance. That response then leads to the 4th step, which is to destroy or suppress the invader. Now we've talked about our immune responses and what the function is. Let's just take a quick moment to then talk about the different parts. So I said the first part is the lymphoid. So lymphoid tissue is where our immune cells form and mature. The main gland associated with our lymphoid is going to be the thymus. And the thymus is where we produce our T lymphocytes. The other primary lymphoid tissue is going to be our bones. So in the bone marrow is where we have a lot of our immune cells mature and develop. Okay, so thymus, bones make up the primary lymphoid tissue. The secondary lymphoid tissue is where we then store mature immune cells and where they interact with pathogens and initiate that immune response. And so this occurs in our lymph nodes that we have throughout the body. It occurs in our spleen. We have it in what we call the diffuse lymphoid tissue, so things like our tonsils, our GALT, which is the gut-associated lymphoid tissue. And then there's clusters that are spread all throughout the body of this lymphoid tissue. And this is, again, where the mature cells kind of interact and wait for pathogens. That's the lymphoid tissue. I said the second part is the actual immune cells. Immune cells are called leukocytes. Sometimes we refer to them as white blood cells. And we can group them by their morphology and their function. So there's 2 main morphologies. There's the types of cells that we call granulocytes. And we name them this because when you look at these cells under a microscope, they're cytoplasm. They contain granules, and so you see these little, like almost crystal-like structures in them. Okay, the granulocytes are designated in their names with the phils. So this would be our basophils, our eosinophils, and our neutrophils. The other morphology that we have is what we call the antigen-presenting cells, or APCs. Okay, these are our monocytes, our lymphocytes, and our dendritic cell. And what they do is these guys, they display fragments of foreign proteins on the surface of their cells. I'll explain that in just a minute. Okay, the function, if we group them by function, it's really how they go about destroying pathogens. So we have those cells that use phagocytosis, in which they swallow up invading cells. Okay, these would be our neutrophils and our monocytes. And then we have the cytotoxic. These are ones that produce a chemical toxin that destroys the cells, that destroys the pathogens. These would be our eosinophils and some of our lymphocytes. Not all of them, but a handful of them. So now let's talk about the different types of cells found in the immune system. This table is taken from your book. It does a great job of explaining the different types, their classifications based off of morphology and function, whether they're circulated or not, and then finally their primary functions. So let's begin with basophils. Basophils are not circulated in the blood. They're found primarily in the skin, lungs, and GI tract, the parts of the body that come in contact with the external environment. And they mediate allergic responses. So they tend to release histamine and heparin that mediate that inflammation and deal with an allergic response. So if we're going to use an analogy for all of our cells to kind of describe them, let's use an analogy of an army. So the basophils are kind of the watchtowers, right? These are the sentries that you put out, and they look for incoming allergens that could be potentially damaging to our army. Eosinophils, they also play a role in this, but rather than being fixed like the basophils, these ones are kind of moving around. They are found in the GI tract, again, in our connective tissue, lungs, urinary tracts. And eosinophils, they tend to fight off parasites that have moved into our body and are leeching off our resources. Okay, in our army analogy, the eosinophils would be the scouts. Okay, not just scouting ahead of what's going in there, but they're also going through their own ranks and making sure, hey, has any enemy gotten in? Is there somebody that is being parasitic? That's the eosinophil. Our neutrophils, these make up, now they're a phagocytic cell, and they engulf bacteria that enters into the body. So they can only eat maybe at most 20 bacteria, so they're dispendable, right? That's like our frontline infantry. We send these out at the first wave of an encounter in our innate immune response, and they don't live very long, right? 1 to 2 days. They're kind of like, your Marines. You just send, you say, go, kill. But they're not as efficient at killing as some of our other cells. So the next class is our monocytes. So monocytes are the precursor to a macrophage, and we find them in our blood. Then the macrophages move into larger tissues and they kind of patrol areas. And you can think about these like our commando killers, right? These are the special forces. They also engulf bacteria and pathogens, but they are much more efficient than our Marines. They can engulf more than 100 bacteria. They're the ultimate killer in our army. So again, you can think of special forces of going out. On top of that, they contain antigen-presenting cells, which means that they can recognize pathogens that they have fought before. How do they recognize them? By taking trophies. Literally, they cut off parts of the body of the pathogens that they destroy, and they stick them on themselves like trophies, and that's how they can recognize those things. So, yeah, kind of gruesome in my analogy, but it's like these people are going off and they're cutting off, they're taking bones, they're taking skulls, they're taking those things as trophies to recognize, hey, these are the enemy, and when I see one of these, I'm going to kill it. Next class of cells are our lymphocytes. So lymphocytes, they get circulated in the blood, and then they're associated with the acquired immune response. Even though they get circulated, most of our lymphocytes don't stay in the blood. They move into the lymph tissue. And there's 3 classes of lymphocytes. We have our B lymphocytes, and that includes our plasma cells and memory cells. We have our T lymphocytes, so these are our T cells, cytotoxic, and our helper T cells. And then we have the third class, which is our natural killer cells. And lymphocytes play a couple of different roles, right? I said they're associated with the acquired immune response, but the B cells, these act as what we call effector cells. So when they get activated, they either become a memory cell or a plasma cell. Plasma cells, their whole function is to secrete antibodies. And antibodies serve as a mechanism for other antigen-presenting cells to recognize the enemy and attack. So if you think about this, these are kind of like our generals, right, of the different armed forces. They're the ones that say, hey, look, we're giving you the info. We're working in intelligence. They're intelligence officers. And they say, hey, this is what we should expect to find. And they're sending out, instead of trophies this time, they're sending out pictures of saying, if you see this person, attack. Okay, that's the plasma cell. They're the ones that are sending out those pictures. Now, the memory cells are in the different platoons. These are the ones that actually say, look, the enemy's over here. Let's go attack. So they're the leader of, they'd be implanted with our macrophages and with our dendritic cells and our T cells. And they say, yep, come on, we need to engage over here. They're the Army intelligence that's on the ground in the battalions. Now, our T cells can be one of two classes. So the cytotoxic and the helper T cells. The cytotoxic T cells, they use, again, poison to kill their prey, and they tend to focus on virus-infected cells. So again, a highly trained intelligence officer that is looking at people within their own army that have been infected by the enemy. And they say, hey, look, no, this one's been infected with the virus. We need to destroy this cell. So the helper cells, they can bind to other immune cells that have a MHC complex on there. And then when they bind, they kind of like make that cell, they energize it, soup it up, and then they usually kill whatever infected virus cell that then they need to. Finally, we have the natural killer cells. Natural killer cells are the cells that deal with, say, like cancer and other things. These in our army are the secret police that are going around and saying, has someone turned to the enemy? Has one of our own become radicalized and no longer functioning? When they find those ones, they kill them. The last type of cells are our dendritic cells. These are found in our skin and in some of our organs. They're not circulated in the blood at all. And they, again, have that antigen presenting cell. So when they collect or capture antigens, what they do is they migrate to the lymphoid tissue, and then they present those antigens to lymphocytes. So these are our spies. We send them out, they detect the enemy, gather the intelligence, and pass it on to then our generals on the ground and to our commandos, our special forces, to then be more efficient at killing them. They gather that intelligence directly from the enemy themselves. So those are the different types of cells. Let's look at what happens when some of these cells bind and what causes them to destroy. So in this first one, we're looking at a pathogen. It's going to be more of an innate response. So on this particular, let's say it's a neutrophil, right? It has phagocyte receptors that bind to the pathogen itself. So there's usually sequences in the pathogen that then bind onto these sites. Once it binds, that triggers the phagocytosis. It brings it in. once it's inside the neutrophil, it'll use a lysosome with enzymes to digest that pathogen and destroy it. Okay, after it has destroyed, if it is an antigen-presenting macrophage, right, it can then break it down. It gets these little digested antigens, and it can attach it onto its own receptors to make it specific for that type of pathogen the next time it encounters it. Okay, so again, when we were talking about our macrophages and how they take trophies, once they learn to kill something, that's what they say, hey, this is what I'm good at, I'm going to keep killing that, and I put those antigens on there. Now, if we have a bacteria that has a capsule, that makes it difficult for the phagocytic receptors to just bind to it. So now we need to have membrane receptors that are able to get through and somehow bind onto it. And the way we use that, the way we encapsulate these bacteria that have capsules is we utilize antibodies. So antibody molecules, these are being produced by our plasma cells. They bind on to the polysaccharide capsule, and then we have recognition sites for those antibodies. And the cell, when it binds in, same thing, it brings it in, it digests it. We can then take the antigens from that one, put it on, and we'll be better at identifying them in the future. Let's talk about our innate immune response. So that innate response is going to occur. It's the first line of defense after we've passed over these physical and chemical barriers. We've gotten past the skin and the mucous membranes. And so in this response, we're going to have basically our phagocytes ingesting foreign material. So they recognize the PAMPs that are found on the bacteria. Okay, these are these toll-like receptors. This is what they bind to. They can use the chemotaxins to kind of identify them. And yeah, they're going to then bind and destroy them. Other things that we find with the innate response, things like pus. This is where we have a lot of white blood cells that are being excreted, and they're leaving the blood vessel for the tissue. That's why we find pus. We also have opsonins, which are antibodies and plasma proteins that are then ingested. Okay, also in the innate response, we'll have our natural killer cells. These, again, are fighting off those like tumor type cells. And it's usually directed by cytokines, which are a protein that initiate that inflammatory response. Let me speak a little bit further about natural killer cells. So again, I said these were the secret police that are identifying their own troops that have turned. What we mean by that is really they can target two types of things, either virus-infected cells or they can target tumor cells. So the virus-infected cells, what they do is when they find them, they basically induce apoptosis, which is cellular death. So just like the secret police, they don't necessarily do the killing themselves. They find those people, they bring them into a dark room, and they say, all right, you got two options. Option one, you kill yourself in a less painful manner. Option two, it's going to be really, really painful if we have to do it. And people opt to do that themselves. Okay, that's me extrapolating beyond and anthropomorphizing cells. But essentially, that's what they do. They brainwash the cell into thinking it needs to die, and the cell kills itself. Some natural killer cells will attack tumor cells that have started dividing unnaturally. Same thing, using apoptosis. They can also secrete antiviral cytokines. These include what we call interferon. So interferon is a cytokine that seeks to try to identify cells infected by viruses and prevent those viruses from replicating. So we have alpha and beta interferons, and they stop the cell from going into the cell cycle so the virus can't be copied. I've spoken about cytokines a couple times now, and let's define them a little bit more clearly. These are proteins that create an inflammatory response. And inflammation, that's the hallmark of innate immunity, right? That's where we get the swelling, the redness, the increase in temperature. And the point of that, when we cause that inflammation, what we're doing is we're attracting other immune cells and chemical mediators to the site. So those first line defenders, if they start to fight, they start that swelling, they start that inflammation to basically say the battle is over here. We've engaged the enemy. Secondly, what it does is it produces a physical barrier that cuts off forms of retreat of the enemy, right? It makes sure that the infection doesn't spread. That's why you get pus and other things that are forming in that to prevent the area of spreading out and going further into the body. And finally, the other aspect of inflammation is it promotes tissue repair. So even once we get the infection under control, we still need to fix things, and we've brought blood and other aspects to this area so that we can start. Okay, so we talked about innate immune response. Now let's talk about the acquired immunity. This is the antigen-specific responses. Everything about the adaptive immune response is specific. We want to be able to specifically and accurately recognize pathogens, especially difficult to recognize pathogens, ones that have encapsulated themselves. The way this is accomplished is that when the body is exposed to a pathogen, it starts to produce antibodies. So as we start to engulf a few of these, we start to learn about them, we start putting those antigens, and we start making antibodies that then recognize those pathogens. Okay, a natural infection, we get natural antibodies, or like in the case of getting a vaccination, you're creating artificial antibodies to help recognize those pathogens. So we start first with a passive immunity that occurs when we acquire the antibodies made by another organism. This everybody went through at some stage in their life. When you were, if you were being breastfed, you got passive immunity from your mother. When you were a baby in the womb, you got passive immunity through the placenta. The antibodies of your mother was shared with the baby. We can also hit what we call artificial passive immunity, where we can take gamma globulins from one person and give it to another person. The cells associated with acquired immunity are going to be our lymphocytes, specifically our B lymphocytes. Now, B lymphocytes, like I said, they become plasma cells or memory cells. And when we get a mature, we call it naive B lymphocyte, okay, we stick an antibody into their cell membrane, and as soon as that antibody gets put on the cell membrane, it becomes a surface receptor, and it then starts making clones of itself. And what we get is clones then start to respond to antigens, and it differentiates itself into what we call an effector cell. And then we can get memory cells that then remember that, and we can have clonal expansion. On the next slide, I will show you that chain of events occurring. Okay, so effector cells turn into plasma cells, and the purpose of a plasma cell is to do nothing but secrete antibodies. Okay, this takes a little bit of time, so let's look again. So here are a group of clones. They all have different antibodies. So clone 1 has this U-shaped one. Here's like a box-shaped, and then a V-shaped, representing 3 different types of antibodies. So what happens is when one of these clones comes in contact with a pathogen that has that antigen, it undergoes what we call clonal expansion. Okay, it starts replicating itself like crazy because it says, uh-oh, we have a foreign invader. There's probably more of them. Let's start dividing. So we then have these effector cells. And these then can turn into plasma cells where they do nothing but produce antibodies. Some of them will turn into memory cells. And the purpose of a memory cell is to live a long time. These are short-lived. Memory cells stay in the body for a very long time and says, okay, even after this fight, we recognize that the enemy is going to come back, and so we will wait. And so what we see is after that first antigen exposure, it generally takes about 2 weeks to start generating a lot of antibodies. Now, some of you may be recognizing this right now with the COVID vaccinations. Technically, after you get your first vaccination, they tell you it's going to take about 2 weeks to 3 weeks to start producing antibodies. After your second one, it's going to be even faster. But again, they say 2 weeks after the second dose, you're good. Now, what happens after that first response? Okay, so the first wave came and went. Let's say now later, you then encounter that antigen again. Who's going to encounter it? The memory cell. The memory cell will encounter the antigen, and it undergoes another aspect of clonal expansion. Okay, so the clones expand, but this time they expand much more rapidly. And we create additional effector cells and plasma cells, and they start secreting antibodies. But now it's a lot faster. So you see after that second exposure, we start getting a huge secondary immune response, in a matter of days afterwards. Antibodies are made up of 5 different classes, and you need to know these. So we have the IG stands for immunoglobulin. So the 5 different classes. The first class is G. This is the most abundant that we have if you take your plasma serum, okay? We produce this in the secondary responses. Okay, so not the primary response, but the secondary response. These are the ones, again, in the COVID, in the era of COVID, these are the ones that we measure when we do an antibody test to see, hey, do you have G antibodies? Okay, A, we tend to find in external secretions. So you would get this in like mother's milk. You tend to get some of the A antibodies. E, these are antibodies. They target gut parasites, and they're also associated with our allergic responses. M, so these are the ones we get with our primary response. They react strongly with complements, but they're not nearly as fast. So you start out first making M antibodies, and then on the second exposure, you would make G antibodies. And then D, we don't quite fully understand what D antibodies are, other than to say that we find these ones, they tend to be attached onto our B cells. Okay, where do these go? We've talked about our beta lymphocytes. Now let's talk about our T cells. So T cells, they're stemming from the same multipotent stem cell found in the bone marrow. We start to get the precursor. T cell will then move to the thymus, and it's in the thymus that it will then mature. It'll either become a cytotoxic T cell or it'll become a helper T cell. The helpers, once it binds to an antigen presenting cell, so this is probably another immune cell that already has that antigen, it'll help it fight. It teams up, activates other immune cells to fight. The cytotoxic T cells will find usually self cells that already have these types of MHC targets on there, bind to them, and they will kill them. Generally, these cells are already infected with a virus. Now, what is an antibody? Your book goes into great detail on the function and the structure of an antibody. I just briefly want to talk about this. It's made up of 4 peptide chains, okay, 2 of which we call are heavy, so they're longer, and then we have these light chains. And they kind of look like little Ys, right? They're identical mirror images of itself. And what we find is at these we have these antigen binding sites, and they become really highly variable. The sequences that make these up are really, really variable. And so just small changes. You can almost think about like a lock and key, right? This is the lock, and we can kind of change based off of what amino acids we stick in here. We can change the shape of what type of keys can go in. The keys would be the antigens. Okay, and so what they do, they bind on to our bacterial toxins, right? They bind onto them, and then we have enhanced phagocytosis where we can kind of bring them in and then destroy them. So let's look at two responses. I'm not going to necessarily test over this, but again, just to summarize everything to show you why this all matters, let's look at what happens when bacteria enters into our system. So when bacteria enters in, the first thing that's going to do is it's going to activate our complement system. Okay, so we're going to start forming a membrane attack complex that surrounds these guys. So we have our mast cells that start producing our chemotaxins. We start producing our opsinogens to help us engulf these. Okay, so we're making chemotaxins. We start releasing histamine. That histamine makes it easier for other white blood cells to move out of the capillaries and come in. That's what then happens is we start seeing the antibodies and complements from other proteins start to enhance the phagocytosis. So we're bringing our lymphocytes in. We're then coating them with opsinogens that are going onto the bacteria. So the bacteria get coated by our opsinogens. We start activating and putting perhaps antibodies onto our beta lymphocytes, and that allows us to start attacking and killing them. Okay, at that point, that's our innate response. We'll then also start causing an acquired immune response. So the beta lymphocytes start turning into our helper cells, our plasma cells to secrete more antibodies in case this bacteria comes back. We start getting our helper cells that start, developing so that if we have a longer infection, they can team up with our macrophages and help have a better response. If we have a virus, again, virus comes in, it invades. We start to produce antibodies that act like kind of an opsinogen. They cover the virus, and that helps our macrophages digest the virus and kill it. Once we've killed a few, we start making antigens and putting those on. That makes us better at targeting these. Additionally, we start secreting cytokines that bring in other white blood cells to help in the fight. They start creating things like interferons. Okay, we start putting these viral antigens onto infected host cells. Helper T cells start pairing up with some of our macrophages to be even better. We start creating more antibodies through our beta lymphocytes that turn into plasma cells that again help us with this initial infection if it happens again. Once we have a helper T cell, it might become a cytotoxic T cell, and then it can bind on to these cells that have the virus in there that we've identified and tagged with the virus and help destroy those cells by telling them to kill themselves through apoptosis.